Dear students, a warm welcome to you for today's lecture entitled General Structure of Viruses. In this lecture, you will be able to know the brief history of viruses, to understand what are viruses, to know the structural organization of viruses, to know the symmetry of virus particles, to know the shape and size of different viruses, to understand the variation in the genomes of various virus groups, and to study the tailed structure of polyhedral, helical, and complex viruses. The history of viruses dates back to 1670 when the color breaking of tulips was published in Trait des Tulipes, wherein it was mentioned that the color variegation might be due to a disease. The first scientific proof of the mosaic disease of tobacco came in 1892 when Dmitry Ivanovsky proved that the infected sap was capable of inducing the mosaic disease in healthy plants, even after passing through bacterial-proof filter candles. M.W. Beijering in 1898 gave the name to this infectious agent as Contagium vivum fluidum, and later on as filtrable viruses. Meanwhile, Bohr and Beijering coined the term viruses for this infectious agent. The structural virology started only after the work of Stanley, who in 1935 isolated the tobacco mosaic virus in crystalline form, for which he was awarded the Nobel Prize. With the advancement of optical instrumentation, structural and molecular virology gained momentum. Now, characters of viruses. Loff and Turner in 1966 described the following characters of viruses: that viruses possess only one type of nucleic acids, either DNA or RNA, and never both. Virions reproduce from their sole nucleic acid. Virions are unable to grow or undergo binary fission. They lack the genetic information for the synthesis of Lippmann system. And viruses make use of their host machinery, that is absolute parasitism. However, F.C. Bowden reduced the long list of characters, defined viruses as submicroscopic infective entities that multiply only intracellularly and are potentially pathogenic. Hehon in 1964 called these definitions as orthodox and treated viruses as transmitters or vehicles of information-bearing genetic material, and defined viruses as bits of infectious heredity in search of chromosomes. This definition seems to be relevant when the second school of thought, that is retrogressive evolution of viruses, is taken into account. Now, general structure. Generally, a virus consists of a strand of nucleic acid surrounded by a protein coat. Hence, viruses are nucleoproteinaceous. The whole virus particle is called virion. The protein coat is known as capsid, and the individual protein subunits are called capsomeres. The nucleic acid together with protein coat is called nucleocapsid. Therefore, viruses have three important components which give them the structural organization and help in its replication in an appropriate host cell. These components are capsid, genome, envelope, and enzymes. Capsid, it is a protein covering which encloses the nucleic acid. Hence, capsid along with the nucleic acid is called nucleocapsid. The proteins in the capsid are arranged in smaller identical subunits. Each subunit is called a capsomere. The arrangement of capsomeres is characteristic for a particular virus. The capsomeres are made up of monomers called protomers. There may be similar or several types of protomers in the capsid. The protomers are connected to one another with the help of bonds. Protomers as well as capsomeres, once exposed to a proper condition, associate spontaneously to form the capsid without any external help. This process is called self-assembly. For example, in tobacco mosaic virus, protomers aggregate to form a two-layered disc. It is followed by the association of code proteins with RNA at a special site near three ends of the genome. RNA forms a loop and inserts itself inside the hole of the disc. The association of protomers continues till the whole of the RNA gets inserted in the capsid. The arrangement of capsomeres in association with the nucleic acid gives a definite symmetry to the virus particle. It may be helical or polyhedral and sometimes represents both. The capsid performs following functions in a virus particle. Number one, it protects the nucleic acid from any kind of damage as a result of mechanical force, ultraviolet radiations, or due to the activity of host enzymes. Number second, it helps the virus in its attachment to a host cell with specific receptors. It enables the virus particle to penetrate the cell membrane of the host to inject the nucleic acid in the host cytoplasm. Now, genome. Unlike other organisms, viruses show a great variation in their genome. Primarily, viruses possess either DNA or RNA as their genetic material, but never both. It may be single-stranded or double-stranded. It can be linear, circular, or segmented. The virus genome varies in size from 3,500 nucleotides to about 230 kilobase pairs. With the advancement in the molecular biology techniques, attention has been diverted to explore the genomic composition of viruses. The following generalizations may be made about the viral genome. Plant viruses usually contain RNA. It may be single-stranded or double-stranded. Some plant viruses contain DNA. Animal viruses generally contain double-stranded DNA. However, some animal viruses possess single-stranded RNA and a few have double-stranded RNA. Bacterial viruses or bacteriophages contain DNA which may be single-stranded or double-stranded, or they possess RNA which again may be single-stranded or double-stranded. Now, RNA virus genome. RNA genome is of two types, that is single-stranded RNA and double-stranded RNA. A single-stranded RNA genome may be of plus sense, that is with the same polarity as that of messenger RNA, negative sense, which is incapable of gene expression, or ambisense, that is a mixture of both plus and minus sense. Now, let us take positive sense or plus sense single-stranded RNA. The RNA of such viruses, after uncoating in the cytoplasm, acts as messenger RNA and directs the synthesis of virus proteins responsible for genome replication. Virus groups with such genomes share some common features. For example, number one, purified plus sense RNA can induce infection directly without any virus protein. Number second, virus genome at its 5' and 3' possess an untranslated region which do not code for any protein. Number third, both the ends are modified, 5' by a methylated nucleotide cap and 3' by polyadenylation. The modification helps the viral RNA in its recognition by the host cell. The examples of single-stranded plus sense RNA viruses are caliciviruses, astroviruses, and picornaviruses, tobamoviruses, and bromoviruses. Now, negative sense or negative strand RNA viruses. After uncoating of virus particle in the cytoplasm, the negative sense RNA strand remains associated with the transcriptase enzyme to synthesize a messenger RNA which codes for structural proteins and enzymes that direct the synthesis of plus RNA. From plus RNA, more negative RNA strands are synthesized which act as genome for progeny viruses. Rhabdoviruses, paramyxoviruses and orthomyxoviruses have minus sense or negative sense RNA. Rabies virus, tomato spotted wilt virus and potato yellow dwarf viruses are the common examples. Now, ambisense genome organization. Some viruses are partly plus sense and partly minus sense. Therefore, they represent both types of genomes. For example, RNA viruses and some Bunyan viruses are the examples of ambisense viral genome viruses. Now, double-stranded RNA virus genome. In case of animals, only reoviruses possess double-stranded RNA, while as in case of plants, Potiviridae and Reoviridae possess double-stranded RNA. Wound tumor and rice dwarf viruses are the common examples. DNA virus genome. On the basis of the genome size, the DNA genome of viruses can be divided into two groups: smaller DNA genome and larger DNA genome. Smaller DNA genome, the best studied example of the smaller DNA genome is that of bacteriophage M3. The genome of this virus is circular with a single-stranded DNA and has about 7,200 nucleotides. The double-stranded DNA genome of smaller size is that of phage lambda with 49 kilobase pairs and T4 phage with a genome size of 160 kilobase pairs. The DNA is of linear shape. In case of animal viruses, parvovirus and polyomaviruses have smaller DNA genome. However, parvovirus is the only single-stranded DNA virus. Now, larger DNA genome. There is a large number of viruses whose genome is double-stranded and considerably large. The genome of these viruses resembles that of the host cellular genome in many respects, such as presence of histone proteins, polyadenylation, and in presence of split genes through coding and non-coding segments. Herpes virus and adenoviruses are the examples of viruses with large DNA genomes. Herpes virus has a genome size of about 230 kilobase pairs with a linear double-stranded DNA, while as that of adenovirus is 30 to 38 kilobase pairs in a linear form. Segmented and multipartite genome. Dear students, when the genome is divided in two or more nucleic acid molecules and is packed into a single virus particle, the genome is said to be segmented. Orthomyxoviruses, reoviruses, and Bunyan viruses are the examples of segmented virus genomes. Influenza virus has 8 RNA molecules with 890 to 2341 nucleotides. In case the genome is segmented but packed into separate virus particles, it is said to be multipartite. Such viruses can have 2 or 3 capsids and are accordingly called bipartite and tripartite viruses. Multipartite genome is only found in plant viruses. For example, Gemini viruses. In these viruses, genome is packed into many capsids. Now, third part of the virus particle is envelope and sometimes enzymes. Many viruses, those of plants, animals, and bacteria, have complex membrane structures around the nucleocapsid, which is called envelope or papillae. The virus envelope consists of a lipid bilayer with glycoproteins embedded in it. Besides, there are carbohydrates. The lipid and carbohydrates are derived from the membranes of the host cell, whereas the glycoproteins are of viral origin. Envelope sometimes gives rise projections which are called spikes or papillomers. These structures help the virus in the attachment with the host cell surface. Besides, they are used as taxonomic tool in virus classification. Because of flexible nature of envelope, enveloped viruses have a variable shape and are called pleomorphic. However, in some viruses, the envelope is firmly attached to the capsid and the viruses have a constant shape. Based on the presence and absence of envelope, the viruses are grouped as enveloped and non-enveloped viruses. DNA animal viruses are both non-enveloped as well as enveloped. Non-enveloped viruses are parvovirus having single-stranded DNA, papovirus with double-stranded DNA, adenovirus with double-stranded DNA, and iridovirus with double-stranded DNA. In case of enveloped viruses, hepadenovirus is partially double-stranded with DNA as it is genome, and herpes virus with double-stranded DNA. RNA animal viruses, they too are non-enveloped as well as enveloped. In non-enveloped, picornavirus has single-stranded RNA, whereas reovirus, they have double-stranded RNA. In case of enveloped RNA viruses, rhabdovirus, they have single-stranded RNA, togavirus with single-stranded RNA, and orthomyxovirus with single-stranded RNA, Bunyan viruses with single-stranded RNA, coronavirus with single-stranded RNA, arenavirus, retrovirus, and paramyxoviruses have single-stranded RNA. Influenza virus is a well-studied example of an enveloped virus. Spikes emerge out 10 nanometers from the surface at about 7 to 8 nanometer intervals. These spikes have enzymatic, absorptive, heme agglutination, and or antigenic activity. There are two types of spikes present in influenza virus. These are called H spikes and N spikes. N spikes possess an enzyme called neuraminidase, hence the name N spike, which helps the virus in penetrating the mucous layers of the respiratory epithelium to reach the host cell. Besides, neuraminidase helps in assembly process and budding out of virus from the host. Other spikes with heme agglutinin proteins bind the virus to red blood cell membranes and cause heme agglutination. Usually, viruses lack enzymes, but still some viruses do have enzymes in their capsid or envelope. For example, neuraminidase in influenza virus. Some enzymes are involved in nucleic acid replication. For example, RNA-dependent RNA polymerase in influenza virus and RNA-dependent DNA polymerase in human immunodeficiency virus or HIV. Now, symmetry of viruses. Dear students, symmetry refers to any structure which when rotated around an axis so that the same form is seen from all sides. The morphology of virus is determined by the arrangement of protomers, which are protein morphological units. Primarily, viruses have two kinds of symmetries, namely spherical or icosahedral and helical or rod-shaped. However, some viruses have binary symmetry, that is, they are partly helical and partly icosahedral. Therefore, viruses occur in three main shapes, vis-a-vis icosahedral, helical, and complex. Now, icosahedral or polyhedral symmetry. When protomers aggregate into units of 5 or 6, they condense to form an icosahedron with 20 faces of equilateral triangles and 20 apices. Pentamers or pentones are at the vertices of the icosahedrons, and hexamers or hexones form edges and triangular faces. Viruses with such a type of symmetry are called icosahedral or polyhedral. The number of capsomers in an icosahedron varies from virus to virus. For example, 5X174 has 12 capsomers, turnip yellow mosaic and polio viruses have 32 each, polyoma virus has 72, herpes virus has 162, and adenoviruses have 252 capsomers. A common example of an icosahedron is the adenovirus, whose detailed structure is discussed here. The adenoviruses are DNA containing viruses which cause mild respiratory infections in humans. Some adenoviruses cause tumors in animals. The virus has a diameter of 75 nanometers and its coat consists of 11 to 15 distinct proteins. The major coat proteins are called hexons and constitute the major proteins of the faces of the icosahedrons. The hexons are actually trimers composed of 3 identical polypeptides. At the 5 vertices of the icosahedrons are 12 pentones. Some characteristic fibers are attached to the pentones which help the virus particle in its attachment with the host cell. The molecular weight of the DNA is 20 into 10 raised to power 6 Daltons. Now, helical symmetry. When the protomers are arranged in such a way that they form a long, rigid, hollow tube, the virus symmetry is said to be helical. Tobacco mosaic virus or TMV is the best studied example of helical capsid. TMV is an RNA virus about 150 angstrom units thick and 3000 angstrom units long, with a central hole of 40 angstrom units. The capsid is composed of 2130 identical protein subunits with a molecular weight of 17,000 Daltons. Each subunit has 150 amino acids. The helix has 16 and 1/3 subunits per turn. There are 130 turns along the helix. The length of the helical viruses is determined by the length of the nucleic acid. The nucleic acid in TMV is a single-stranded RNA molecule coiled into a helix and a diameter of 80 angstrom units. RNA helix has 49 nucleotides with a pitch of 23 angstrom units. There are 6,400 nucleotides with a molecular weight of 2 into 10 raised to power 6 Daltons. Now, complex symmetry. Some viruses do not fit into the helical or icosahedral symmetries, but rather show a combination of both and therefore have a binary symmetry. Pox viruses and bacteriophages are the common examples of complex viruses. The structure of bacteriophage T4 will be discussed here as an example. The bacteriophage is tadpole-like with a polyhedral head and a cylindrical tail. The head is an icosahedron elongated by 1 or 2 rows of hexamers. In the middle, it has a diameter of 96 into 65 nanometers and is made up of 2000 similar protein subunits and contains a circular double-stranded DNA about 53 microns long. The tail has helical symmetry. It is attached with the head by means of a collar or neck piece. It has a central hollow tube, a sheath surrounding the tube, and a complex base plate. The sheath is made up of 144 protein subunits arranged in 24 rings, and each ring made of 6 protein subunits. The base plate is hexagonal and has a pin and a jointed tail fiber at each corner. The length of the tail fiber is 130 angstrom units. The tail fibers are the organs of attachment to the specific sites of the host cell. Dear students, therefore, we had cursory glances over the characters of viruses, the general structure of viruses, and the symmetry of viruses. Therefore, we conclude this topic here. Thank you.